Assalamu alaikum. On behalf of Ahsan Embryology, let me welcome you to a series of presentations on embryology. This is the first introductory presentation. So in this introductory part, I'll try to discuss the initial conception on the embryology, the significance of its study, and some terminologies, including the basic idea on cell division. So, what is actually embryology? What does embryology means? It's a study of embryo, the beginning of the human or living organism. It starts from a single cell and gradually it forms the total organism, multicellular organism. So this is the study of formation of this organism from one cell stage, zygote. In case of human, this is known as human embryology. So human embryology will discuss the human body formation from a single cell. The embryology study will help the doctor to have the knowledge on the various relations of the anatomical relations of the structures, body organs, will help the doctor to explain the various processes that are occurring in the human body. This will help the doctor to develop new techniques for the diagnosis of certain problem prenatally, means before the birth, and take appropriate measures so that deformity cannot develop. It helps the doctor to decide about the prevention or control or combat with the infertility. This will help the doctor to counsel the couple to maintain a healthy reproductive life, and by this they will be able to get a healthy baby as an outcome. Now let us discuss some basic terminologies. Here you can see the arrow is showing the cell. It is indicating that one cell was there and that repeatedly divides to make more. This is known as proliferation. So proliferation means multiplication leading to increase in number of that substance. If it is cell, then it is proliferation of the cell. If it is bacteria, proliferation of the bacteria. So in this way, that increasing by multiplication is proliferation. What is growth? Growth is specific terminology that means positive increase in size, means because of the proliferation, the size of the organ will increase, size of the tissue will increase, and subsequently the body will increase in size. It is always controlled and regulated in every organism. So in human body, you see the liver will get the shape because of the proliferation of the cell. After getting shape, this will increase gradually, and gradually the rate of the increase will be reduced, and at some time the size will attain the maximum size, and uh, growth will stop. Only repair will continue. Differentiation is another terminology. Frequently you will get it in embryology. Look at the figure. There are green apples, one is red. Suppose this green apple, because of some effect of some chemical substance, the same tree has started growing this or giving this type of apple that is different in color, but it is still apple. So this is simply differentiation, change to another distinguished form. Easily you can identify this may have different taste, may have different function, may have different effects. So cell from one stage or one moves to another state with different functions. This is differentiation. Like here you can see one cell. Here this is one cell. It's the mother cell. So it can increase in number by proliferation. Suppose this is one cell differentiated to another form. This is another cell, and this is now converted into an epithelial cell covering the surface. The function of this cell is to protect the surface from mechanical, chemical, physical injuries. On the other hand, from this cell, another cell is produced that is changed to another form, that is the neuron. So neuron is doing something else. Uh, in this way, the cell changes from one form to another. This is differentiation, and uh, cell usually converts into a more specialized type from less specialized. Like this is less specialized, and now this is epithelium, so having very specific function. This is less specified. Now it is differentiated into new. Here I am showing one cell. This is having multiple capability. This is not a specific cell with a specific function. This can convert into a fat cell to store fat. This can differentiate into cells that can lead to muscle, and now it can contract and induce movement. This is now converted into epithelium to covering the surface. This is now converted into blood cells. So this one is the mother cell who can differentiate into different cells with a specific function. So this type of cells are known as stem cell, who can change to another specialized function uh, carrying cells. We know this one. This electricity, if passed in this manner over an iron rod, the iron rod will be converted into a magnet. And we used to say this is inducted into a magnet, the process of changing. So here also we'll find that during human development, formation of one tissue or one cell will induce formation or differentiation of group of cells or single cell. So this is known. This process is happening in the human embryo in a series of ways. So here you can see this is the notochord. So over this notochord, usually there is a layer of cell. So notochord induced the cells over it to be changed, and this will go inside the mesoderm in connective tissue type and form tube. Even this junction area is also converted into some other cells that also deep down here to form. This is the neural tube, neural crest cells. So this is induced, this neural tube is induced by notochord. This notochord inducing formation of the neural crest cell, and also some cells will concentrate to form paraxial mesoderm, and changes in the paraxial mesoderm is induced by this neural tube. So the series of process, a group of cells, one cell or a tissue causes another set of cell of tissue to change their fate. That was on the surface. Now this is forming the spinal cord and nervous system. So there are some inducer who induces the cells to respond. Only the responders can respond to this inducer. Other cell cannot. When it is dipping down, other is also coming close to this or chemical substance produced by this, but there will be no change there. So response is limited to some other cells who will be changed in the embryo, and other will not be changed. So inducer has a specific responder, and capable to produce response in the responder is the competence of that capability of that inducer. So inducer should be sufficient competence to influence the responder to be changed. If it is not, there will be some deformity. You are conversant with these figures. You can understand this is diagram of sun, and you can imagine actually who is doing this. So sun is responsible for this crisis, isn't it? So evocator, the factor, the control change. So sun is behind, but who is coming here? This sun is sending something, and that induces some changes. So sun remaining at his position is the organizer and sending some chemical substance. Here you see the heat or radiations, that is evocator, that produces some change in the responder. This is an example. So evocators, factors. This is organization. Organizer is releasing the factors. That factor influencing the responder to produce change. Let us compare. This is the notochord. So notochord is there. Notochord is the organizer. So that should produce some chemical substance who is capable of producing some change or effect over some cells or tissue. So this is evocator. So organizer releasing the evocator that is specific for certain tissue or certain cells. So this evokes specific change on some area on single cell or group of cells. So in this way, the embryological process is continuing. So from one cell, when they multiply, this is replications producing thousands of cells. One of them or group of them changed into organizer, differentiated. Now this organizer started a series of reaction. So that will send evocator to responder to respond. That respond become organizer again, send some evocator to influence other to be changed. That can become organizer again, and a series of reaction continue, and the whole human body is produced. Now we know that cells are there, and cell is the structural and functional unit of the living organism. If we see inside any organ or any part, we'll find that series of cells are there of different types, and they each has structural orientation, specific type. So this is having plasma membrane, protoplasm, nucleus, cytoplasm, and inside that organelles. In the nucleus there are chromosomes. So all are there, and you are conversant with the structure of the cell. And let us continue to discuss those parts which is required for understanding of the basic embryology. So living cells, living organism, the life actually starts with a single cell. So the cell will divide, and this is known as cell division, and by cell division the whole human body is produced. After that, repair is continued. So cell division may be of two types, mitosis and meiosis. And you have probably some idea about the mitosis and meiosis. I am showing two videos so that you understand, or you can revise the mitosis and meiosis. And after that, I will discuss the basic difference between mitosis and meiosis.
In interphase, the genetic material of a cell increases. Prophase. During prophase, The chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into
males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the two daughter cells into full sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over.
Let us discuss the basic difference, and after seeing the cell divisions, I hope both type of cell divisions you have seen with concentration. So there is some difference, basic differences. You see, there is some difference in events. Mitosis is completed in single stage. The division is complete and they will become gradually enter into the cell cycle and become mature cell. But in meiosis, you'll find that there are 2 stages. In one stage, this is divided into 2 cells, then second stage starts, and each cell divides into another 2 pair of cells, so ultimately 4 cells are produced. What is happening in the cell division? So mitosis, one cell produces 2 daughter cells, and both are identical to the parent cells, and both are diploid cells. Means 2n chromosome, 46 chromosomes, 23 pairs of chromosomes, 22 pairs of autosome, and 1 pairs of sex chromosomes are there. In meiosis, what's happening? One diploid cell produces 4 daughter cells, and the new cells are haploid in nature. Means the mother cell was diploid 2n, but now the daughter cells, each of the 4 cells are haploid, means n number of chromosome. Instead of 23 pairs, it is having 23. So 22 autosome and 1 sex chromosome, either X or Y. This is the result of meiosis. So chromosomal splitting in the mitosis, all chromosome splits and move to the opposite pole and produce 1 cells. 2 cells are produced with splitted 1 member from the splitted chromosome. So they are identical. In meiosis, what's happening? Splitting is occurring at the second stage only. So first stage, there is no splitting. So 46 chromosome will be divided into half to 23 chromosomes, will move to one cell, and in the second stage this will split. So 23 will remain because 23 will split and move to the two different cells. So this way the chromosome splitting helping the cell to become haploid from a diploid. This is also called the reduction division. Mitosis is normal division and meiosis is reduction division. And reduction actually occurring in the first stage. Second stage, there is no reduction. Genetic material, you see, in the mitosis, there is no chance of genetic material exchange. But the process of meiosis is very long, and during this process, several events are happening to have change at the micro level, at the very minute level to the component parts of the chromosome level. Future of the produce new offspring, daughter cell. So mitosis producing normal cell, so this can go into division and can produce another two daughter cells. But meiosis, you see, this is a blind end. So one mother cell divides into 4 daughter cells who are haploid and cannot divide again. So this is half cell. The fate of this cell again starts life, otherwise it remains in this form and dies. So this one half cell requires to fuse with another half cell to produce a complete cell. This is fertilization. So if you understand meiosis, you will get the component member of fertilization and you will understand the embryology in a better way. This mitosis is occurring in all diploid cells except some permanent cell who cannot divide. Mitosis even occurs in germ cells. Spermatogonium is the germ cell. Here, the mitosis occurs. Meiosis starts only in the germ cells and specific germ cells. Like primary spermatocytes allow meiosis, and oocytes in the ovary allow meiosis to produce some germ cells. Thank you very much for remaining with the presentation.